In this video, we are going to discuss the transport of the respiratory gases, which are the respiratory gases, the oxygen and the carbon dioxide. And this is a very important topic. So let's talk about oxygen. Oxygen is carried in the blood in two ways. 1.5% is dissolved in the plasma. As you can see, this is a very small fraction. 98.5, it is bound to the hemoglobin, which are present in the RBCs. And in the hemoglobin, they are bound to the iron. The heme in the hemoglobin, it contains the iron, and that iron, it binds oxygen. This is how 98.5% of oxygen is carried in the blood. Now let's see that we know that each hemoglobin molecule, it is composed of 4 polypeptide chains, and each polypeptide chains, it contains a heme group, and each heme group contains one iron molecule, and this iron molecule, it attaches the oxygen. So if there are 4 polypeptide chains, there are 4 heme groups, there are 4 iron molecules. It means that one hemoglobin molecule, it carries 4 molecules of oxygen. Okay, so one hemoglobin molecule carries 4 molecules of oxygen. All right, and then when it is attached to the oxygen, we call it oxyhemoglobin. Oxyhemoglobin, it is the oxidized form of hemoglobin. It is more reddish, and when the oxygen is removed, we call it deoxyhemoglobin. And this is how it is written, HHB. This is reduced form of hemoglobin. Where is this H coming from? We will discuss it more. Let's see here. So this is HHB plus oxygen. When it combines, this H is released, right? So this is the H which is written here. So this is when we will go over it and it will become more clear to you. But right now, this is how oxyhemoglobin is written, and this is how reduced hemoglobin, we write about reduced hemoglobin. And one hemoglobin molecule transports 4 molecules of oxygen. Now, so the loading of the oxygen, it is very interesting. They call it that they cooperate with each other. It is known as cooperativity, although this term is not mentioned in your book, but maybe you will read it somewhere. So when the first oxygen molecule, it attaches to the hemoglobin, the affinity of the hemoglobin increases for the next oxygen molecule. So when the first oxygen molecule, it attaches to the hemoglobin, it changes the shape of the hemoglobin molecule. Okay, the shape of the hemoglobin molecule is changed, and that new shape, it has more affinity for oxygen. And when second molecule of oxygen, it attaches, now there is further shape in the change, and there is more affinity for the third oxygen. So you can say these oxygen molecules, they are cooperating, they are helping each other to bind to the hemoglobin. And similarly, when there are 3 oxygen molecules attached, now this hemoglobin has more affinity for the oxygen compared to this one. So as more oxygen is being attached, there is increased affinity for oxygen in the hemoglobin. Now, up till this stage, we will say that the hemoglobin is partially saturated. Okay, it is not completely saturated. It has either 1 oxygen molecule or 2 oxygen molecule or 3 oxygen molecules. We will say it is partially saturated. This hemoglobin, we will say it is fully saturated. Now, this hemoglobin, even if you increase the oxygen, it has no site to take up that oxygen, right? So when hemoglobin is fully saturated, all the 4 heme groups are bound to the oxygen. Now increasing the oxygen, it will be very, this hemoglobin cannot take up any more oxygen. And similarly, the unloading of one oxygen molecule, it moves this to the other direction. So let me just take the pen and let me just prop it. When the oxygen molecules, they are removed, so one oxygen molecule is removed, then the next stage, it has lesser affinity, so it moves in the opposite direction. So this is loading and unloading of oxygen molecule. Hemoglobin, it binds reversibly to the oxygen. It means that the oxygen can attach to the hemoglobin. It gets attached in the lungs. When it goes to the tissues, the oxygen gets detached. So let me see, let me make this as hemoglobin. The 4 molecules of hemoglobin. I'm sorry, it looks more like a clover flower. And then let's see, it has taken 4 molecules of oxygen. So this blue is like all 4 of them have oxygen, 4 molecules of oxygen. And when this is present in the lungs, the oxygen, it loads to the hemoglobin. But this is not like a very permanent bond or something that now hemoglobin cannot give up the oxygen. When it goes to the tissues, that this oxygen, it gets unloaded. There are certain factors which determine that whether the oxygen will bind to the hemoglobin or is it going to unload from the hemoglobin. So there are certain factors. Those factors are partial pressure of the oxygen. This is very important. Then we have temperature, blood pH, partial pressure of constant carbon dioxide, and then concentration of bisphosphoglycerate. What is this? We will discuss in the next slides. So these are the factors which influence whether hemoglobin will be attached, the oxygen will be attached to the hemoglobin, or is the hemoglobin going to give away the oxygen to the tissues? Is it going to unload? Now let's see the first factor and the most important factor, the partial pressure of the oxygen. We know the partial pressure of the oxygen in the arterial blood is 100 millimeters of mercury. What does and this 100 millimeters of mercury, it means that it is 20 ml of oxygen per 100 ml of blood. So we say 20 volume percent, right? And at this quantity, at this amount, at this partial pressure, the hemoglobin is 98% saturated. And now let's see what happens in the vein. In the veins, the PO2 is 40 millimeters of mercury. We already did that when it comes, the carbon dioxide, it diffuses into the vein. The oxygen, it is low, it is given in the tissues, and the venous blood has oxygen, a concentration of 40 millimeters of mercury, and it makes up to 15 volume percent oxygen. So as you can see, there is not much difference in the venous blood volume percent and the oxygen blood volume percent. So this is and hemoglobin is still 75% saturated in the veins, right? So 75% saturation means that three of the sides of the hemoglobin molecule, the three heme molecules, they carry three oxygen molecules. And so this is known as the venous reserve. So if something goes wrong, the oxygen remaining in the venous blood can still be used, right? So this is how our body keeps a safety margin, a safety net, that even in the veins, there is 75% of the hemoglobin is saturated. This is known as hemoglobin oxygen dissociation curve, and it tells us at a glance how the hemoglobin, it attaches to the oxygen. So this line, it shows the partial pressure of the oxygen, and this shows the saturation of the hemoglobin. So we will come from this point. Let's first start at here. So in the lungs, we know the partial pressure of the oxygen, it is 100 millimeters of mercury, and at this saturation, the hemoglobin, it has 4 molecules of oxygen, it is fully saturated. And then this blood from the lungs, it goes via the left heart to the tissues, and in the tissues the oxygen is given up to the tissue. And now the concentration of, I'm not talking about carbon dioxide yet, so now the concentration of the partial pressure of the oxygen in the venous blood becomes 40 millimeters of Hg. From here it comes here. So at 40 millimeters of Hg, still 75% of the hemoglobin molecule is saturated. So it's still, there are 3 molecules of oxygen which are attached to the hemoglobin molecule. Also, I want you to notice that at around this point, I will say around this point, this is kind of a straight line. So over here, the increase in the partial pressure of the oxygen, it is not affecting the saturation of the hemoglobin that much, because hemoglobin is almost saturated from here to here. And similarly, it goes both ways. If the partial pressure of the oxygen is decreased, it goes this way, or it is increased, it goes this way. The saturation of the hemoglobin will not be affected that much. Now let's start from here. So at 0 partial pressure of oxygen, the hemoglobin, it has no oxygen. Then as the partial pressure of the oxygen moves up, the hemoglobin, it starts taking up oxygen. The oxygen loads to the hemoglobin, and we know as one oxygen molecule attaches to the hemoglobin, it helps the other oxygen molecule to attach to the hemoglobin, because there is a change in the structure of the hemoglobin. which increases its affinity for the second molecule of oxygen, and so on. So as the partial pressure is moving up, the oxygen, it loads to the hemoglobin. When the partial pressure is moving up, it loads the hemoglobin. Oxygen is being loaded to the hemoglobin, and in the venous blood it is around 75%. And when we move towards the arterial blood, as it moves up at around 80, I will say it's almost 100. It's almost like the same as at 100 millimeters of Hg. So this oxygen, as the partial pressure of oxygen moves up, this saturation increases, but at a certain level it becomes a straight line. So around here, the change in partial pressure of oxygen is not going to make a big difference in the loading of oxygen to the hemoglobin, because hemoglobin is already all 100% saturated, 98% saturated. It has all the four molecules of oxygen. So this oxygen hemoglobin curve, it is kind of S-shaped. It is not very straight. If you see, there is a slight curve here, and then it goes here. It has an S-shaped, and this is because of this cooperativity of the of this hemoglobin, that after one molecule, that becomes easier for the next molecule to attach. Because of this property of hemoglobin, of being attached to the oxygen, and this the mutual oxygen hemoglobin thing, it keeps the hemoglobin saturation in a certain range. For example, at sea level, we know the partial pressure of oxygen in the atmosphere, it is normal, it is like what we are used to, it is maximum. So the alveolar partial pressure of oxygen is 100 millimeters of Hg, and the hemoglobin, it is almost 98% saturated. Now, if someone moves to a very high altitude where the partial pressure of oxygen in the atmosphere, it is low, and because of that low partial pressure of oxygen in the atmosphere, the partial pressure of oxygen in the alveolus is also low. But even at this partial pressure, the hemoglobin is almost 95% saturated. So because over here, like I said before, this is a straight line. When the saturation is maximum, then there is a straight line. And now similarly also, this helps that when we are resting, we do not need that much energy is not being used. So it is the same that the arterial blood, that has 98% saturation, and the hemoglobin is attached to the this saturation means that the hemoglobin, it is attached, firmly attached to the um, to the oxygen. The oxygen and hemoglobin, they are attached, the affinity is high. But as we move to the venous side, the affinity, it decreases, so the oxygen unloads. Now consider if someone is doing exercise. During exercise, what happens, that the partial pressure of oxygen, it further decreases, because there is more carbon dioxide that is produced. So the partial pressure of the carbon dioxide is increased, and the partial pressure of the oxygen, it has decreased, and it can decrease up to 20 millimeter Hg. How does this help? Because at this the hemoglobin affinity of hemoglobin for the oxygen, it decreases, and now the hemoglobin is giving away oxygen. So that oxygen is used for the increased metabolic demand by the body when someone is doing exercise. So this was about the partial pressure of the oxygen. Now there are other factors which also affect the the affinity of the hemoglobin for the oxygen. So basically what we have discussed in the oxygen hemoglobin dissociation curve is how the affinity of hemoglobin for oxygen increases and decreases with the increase in the partial pressure of oxygen and a decrease in the partial pressure of oxygen. When the partial pressure of oxygen is more, hemoglobin has more affinity for the oxygen, and it increases with every additional oxygen. And when the partial pressure of the oxygen is low, the affinity of hemoglobin for the oxygen, it decreases, and the hemoglobin, it gives away the oxygen. So, and that happens and in the tissues, because that is where the unloading has to happen. So there are other factors, and these factors are temperature, the blood pH, the blood carbon dioxide, and the concentration of bisphosphoglycerate. What is this? This is a chemical substance which is formed in the metabolism of glucose. Now let's go. Let's go over oxygen hemoglobin dissociation curve. So this curve, it tells us about the affinity of the hemoglobin for the oxygen at different partial pressures of oxygen. And we already said that as the partial pressure of oxygen increases, the affinity of hemoglobin for the oxygen, it increases and more of the hemoglobin is saturated. And as it decreases, as this line moves from here to here, the partial pressure it decreases. So does this line moves from here to here. The affinity of hemoglobin for the oxygen decreases and the oxygen is unloaded. So this is our oxygen hemoglobin dissociation curve. And now let's and it this certain factors, they move it to the right, certain factors they move it to the left. So when it moves to this direction, when this red graph moves in the direction of the green, we say there it has moved to the right, and this causes unloading of the hemoglobin. And let's see that what let's consider 80, and let's see what is happening at 80. So at 80, the hemoglobin constant saturation is this. Now, as these factors increase, this shifts to the right. And these factors, where are they increasing? They are increasing in the tissues. So just imagine in the tissues what is happening. There is more carbon dioxide in the tissue. So the increased carbon dioxide is increasing, is moving this graph to the right. And then there is increased temperature because there is increased metabolism. That increased metabolism is going to increase the temperature. There is more heat, so again the temperature moves it to the right. And then when there is more carbon dioxide, there is more hydrogen ion, so the pH is decreased. So these three factors, if you think about it, you will realize that at the tissues, these are the conditions in the tissue cells. And what is this BPG, bisphosphoglycerate? This is a chemical which is made in the glucose metabolism. So glucose, when it is present in the cell burns the glucose to make energy in the form of ATP. So glucose is converted into pyruvate, and as a byproduct, there is this substance bisphosphoglycerate. This is known as BPG. So also, if glucose is being metabolized in the cell, now you can see that BPG will be increased in the cell. So we can logically think about these factors, that why these factors will be more at the level of the tissues. And what do we need at the level of the tissues? We need the oxygen to be unloaded from the hemoglobin. We need the affinity of the hemoglobin to decrease for the oxygen, and for the oxygen to unload. So these factors are present in the tissues and they help the oxygen to unload. And this graph, it moves towards the right. There is a right shift. Now let's see at 80 what happens at the same when this moves to the right. Now it has moved to the right, and we see the affinity, the saturation of the hemoglobin has decreased. It has come down from this point to this point. So now the hemoglobin is giving away oxygen. And we see that, so we can say that when the graph moves to the right, the oxygen is unloaded. And why is it unloaded? Because of these factors. And these factors, they hemoglobin affinity for oxygen has decreased because of these factors. At this point, the affinity of the hemoglobin for oxygen has decreased. And where does it happen? It happens in the tissues, right? And now, so this is the situation which happens in the tissues. And which are the capillaries present in the tissues? Systemic capillaries. So maybe at some point they have written that in the systemic capillaries, the oxygen is being unloaded from the hemoglobin. So this basically the capillaries which are in the tissues, we call them systemic capillaries, because they come from the systemic arteries, then they divide and we divide and we divide, and we get the systemic capillaries. And let me go over these factors again in the tissues. So there is more carbon dioxide, right, compared to the arterial blood which is present in the systemic capillaries. There is more carbon dioxide in the tissue. and then there is more, when there is more carbon dioxide, there is more hydrogen ion, which decreases the pH. Remember, carbon dioxide, it combines with water and it makes carbonic acid. That carbonic acid, it dissociates into hydrogen ions and bicarbonate, so we get more H, and we get decreased pH at the level of the tissues. And then because metabolism is happening in the tissues, you can remember it like this, that there is increased temperature. Although when we have temperature or fever, that also causes dissociation of the oxygen. But to remember these factors, you can remember that because metabolism is high in the cells, so there is heat production, so the temperature is more. And then this BPG, you can remember it like BPG, you don't have to memorize this name yet. So this is present in the cells. Why is it present in the cells? Because it is a byproduct of glucose metabolism. So all of these factors, they decrease the affinity of hemoglobin for oxygen, and the oxygen, it is unloaded in the systemic capillaries. Okay, this is what is happening in the systemic capillaries, and then those systemic capillaries will change into venules and bring the blood back. Now this is the right shift. Now this graph can also shift to the left. Now let's see at the same what happens when it shifts to the left. What does it mean? So when it is shifting to the left, now let me draw a line from this blue. So this blue line goes up here to here and over here. Now the saturation of the hemoglobin has increased. This is the situation which is present in the lungs. And in the lungs, the affinity for oxygen has increased. And now the oxygen, it wants to keep its the affinity of hemoglobin, I've said it opposite, the affinity of hemoglobin for oxygen has increased, and now the hemoglobin, it wants to keep that oxygen, right? So this is the left shift. So if someone says the hemoglobin curve, the hemoglobin oxygen dissociation curve has shifted to the right, it means we are in this situation. Right now, the affinity of hemoglobin for oxygen has decreased and oxygen is unloading. And if we say that this curve, oxygen hemoglobin dissociation curve, has moved to the left, it means now just consider the situation in the lungs. And over here, all of these things, they are low. So if you remember this, that the increase here, when you go here, when the curve moves to the left, all of these factors, they are low. So this is, now let's move on to the next slide. We went over the summary of that hemoglobin oxygen dissociation curve, that how the factors affect and what are the factors which move it to the right and what are the factors which move it to the left. So over here, this is from your Pearson text. So they have plotted what happens when the carbon dioxide, it increases. The carbon dioxide, when it increases, it shifts the curve to the right. So this purple one, this is under the normal condition, normal arterial carbon dioxide. This is the curve we get. And what happens when the carbon dioxide, it increases? It moves the curve towards the right, as we discussed in the previous slide. So this has moved to the right, and when there is increased carbon dioxide, we know there is increased in hydrogen ions, and so the pH is low. So three of our factors, that when increased carbon dioxide, increased H plus or decreased pH, they are going to shift the curve to the right. And shifting of the curve to the right, it means that the affinity of the hemoglobin for the oxygen is going to decrease. So which we can see over here, that as the carbon dioxide is increasing, this has moved to the right. And when the carbon dioxide, it decreases, it moves to the left. So this is the situation which happens in the lungs. This upper one over here, I will make an arrow over here, that this is when it goes towards the lungs, when the blood is going to the lungs. And then this is the other situation, is when it is coming to the tissues in the systemic capillary. So this curve, it moves up and down as the blood is moving through the lungs and it is coming to the tissues. Over here, they have plotted the effect of the body temperature on the oxygen hemoglobin dissociation curve. So this is what happens. The purple line is at the normal body temperature. What happens? And when the temperature it increases, it shifts the curve to the right, like we already discussed. And when we have fever, this is shifting to this. Now the blood it is giving more oxygen to the tissue, so it can be used because of the increased metabolic need. And you can remember it like this, that at the level of the tissues the temperature is increasing because the heat is being produced because of the metabolism, and as the temperature decreases, it moves towards the left. The curve it shifts to the right, sorry, to the left as the temperature decreases. So this is the effect of temperature on the oxygen hemoglobin dissociation. So this slide basically says whatever we already went over in that chart, that how the temperature and the hydrogen ions and the carbon dioxide and the BPH, when they increase, they shift the curve towards the right, and which causes unloading. And this occurs in the systemic capillaries. Systemic capillaries are the capillaries which are present at the level of the tissues. In the lung, we say pulmonary capillaries. The term will be used, pulmonary capillaries. And similarly, when these factors they decrease, the curve it shifts to the left. There is a mechanism which is known as Bohr effect. What is Bohr effect? Bohr effect is that when partial pressure of carbon dioxide increases, it causes unloading of the oxygen from the hemoglobin. This phenomenon is known as Bohr effect. So increased, like I said before, that the increasing the partial pressure of the oxygen, it increases the affinity of hemoglobin for oxygen. So then, it's similar as an opposite, the partial pressure of carbon dioxide is increased. It causes the hemoglobin to unload the oxygen. This is known as Bohr effect. And Bohr effect, it happens at the level of the capillary blood in the systemic capillaries. Why is it happening there? Because the systemic capillaries, they are in direct contact with the tissues, and the tissue cells, they're burning glucose, they're doing metabolism of glucose to make ATP. And when they burn the glucose, they produce heat. They also produce carbon dioxide. That carbon dioxide is changed into carbonic acid, then it is dissociated into hydrogen ions and bicarbonate, and the hydrogen ions they decrease the pH, and as a byproduct PPG is also produced in the cells during glycolysis, and we know which is also one of the factors which decreases the affinity of hemoglobin for oxygen. And also heat is produced at the level of the cells, and that heat production it also increases the unloading of the oxygen. So this is like the summary of all the factors we went over, but specifically for Bohr effect is associated with partial pressure of carbon dioxide. When the partial pressure of carbon dioxide increases, it causes the hemoglobin to unload the oxygen. We will go over it a little more detail when I start about the transport of the carbon dioxide, but you have to know this term and what it means. How is carbon dioxide transported in the plasma? Carbon dioxide is transported in the blood in three forms. Number one, it is dissolved in the plasma, which is only 7 to 10%. And then it is transported bound to hemoglobin, which is around 20%. The important thing about this point is that the carbon dioxide does not bind to the heme group. In other words, carbon dioxide and oxygen do not bind to the same part in the hemoglobin molecule. They have different binding sites. The seat where the oxygen going to sit, it is separate from the seat where the carbon dioxide is going to sit. They both have their designated spots, their designated seats. The oxygen, it binds to the heme group. To be more specific, it binds to the iron in the heme group. And one hemoglobin has four heme groups and hence four iron molecules. So one hemoglobin molecule carries four molecules of oxygen, whereas carbon dioxide, its spot is different in the hemoglobin molecule. It attaches to the amino acid of the of the globin chain. We know the globin chain, the four globin chains, they are made up of amino acids. So carbon dioxide, it attaches to the amino acid of the globin chain. And when it does so, it is known as, the hemoglobin is known as carbaminohemoglobin. And this is how it is written. So this is very important. I'm repeating it again and again because you have to internalize it, that oxygen and carbon dioxide, they have different spots. They don't have same seats. They have their designated seats. All right. So this is how hemoglobin travels in the blood bound, carbon dioxide travels in the blood bound to the globin part of the hemoglobin. It is 20%. Then 70% of the carbon dioxide, 70%. So the bigger portion of the carbon dioxide, the majority of the carbon dioxide, it is transported in the plasma as bicarbonate ion. Where does this bicarbonate ion comes from? So the same reaction I have been repeating again and again. Carbon dioxide and water, they combine and they make a weak acid known as carbonic acid. This carbonic acid, it dissociates into hydrogen ion and bicarbonate ion. So this is how carbon dioxide is transported. And remember, this reaction, it goes both ways. It goes this way when the carbon dioxide needs to be transported, and it also goes this way. So when it reaches the lungs, it goes this way and carbon dioxide is expired. This conversion of carbon dioxide into bicarbonate, it happens very slowly in the plasma, but it happens with great speed in the RBC. So why? Because RBCs, they have a special enzyme which is known as carbonic anhydrase. This carbonic anhydrase, it reversibly catalyzes this reaction. Please notice the word reversibly. Reversibly means it can go in either direction and the same enzyme it is used. So what happens that this carbon dioxide inside the RBC, it combines with water and then it changes into carbonic acid, then it dissociates into hydrogen ion and bicarbonate ion. Now this bicarbonate ion, when it is formed, it diffuses outside from the RBC into the plasma. So let me make it here. So this is an RBC, and this RBC, it has this bicarbonate ion which has been made and and it has been catalyzed by the enzyme carbonic anhydrase. So now this ion it has a negative charge, as you can see. Now when it moves out, now the cell wants another electrolyte to move in the cell because to keep its electrolyte balance. So when it moves out, there is chloride present in the blood. This chloride, it moves inside the RBC to balance. Chloride is an anion. So this is known as chloride shift. This is referred to as chloride shift. What is chloride shift? That the rush of bicarbonate from the RBC is balanced by the chloride movement into the RBC from the plasma. So this bicarbonate, it is not going to travel in the RBC, it is going to travel in the plasma. So once it is made in the RBC, because RBC has this special enzyme, the process is very rapid inside the RBC. This bicarbonate, it moves outside and chloride moves inside. This is known as chloride shift. You have to know this. Let's go over this diagram and it will summarize for us what we talked about the carbon dioxide and the oxygen transport. Okay, first I'm going to go over the carbon dioxide. So the size of this arrow, the width of this arrow, it is proportional to the amount of carbon dioxide moving here and there, and similarly for this red arrow, which is representing the oxygen. The thickness shows that how much bigger portion of oxygen is going from here to here. Okay, so now let's go over the carbon dioxide. Let's orient ourselves to this figure. This is the tissue cell, right? These tissue cells are producing carbon dioxide, which is going in the interstitial fluid, and they are absorbing oxygen. right? And this is the interstitial fluid. Let's talk about carbon dioxide first. This carbon dioxide is going here, and this is capillary. These cells, these thin endothelial cells, are telling me this is capillary, and this is systemic capillary. Why is this systemic capillary? Because it is at the level of the tissues. If it was at the level of the lungs, it will be pulmonary capillary, right? So don't be orient yourself to these two different terms. So this is systemic capillary. And then this carbon dioxide, a small portion of carbon dioxide, it goes in and it travels in the blood. So this is capillary. Inside capillary there is blood. Blood has plasma and the formed element, so it travels in the plasma in the dissolved form. So this is a small portion, as you can see. And then there is this carbon dioxide. When it comes inside the plasma, it combines with water and it makes carbonic acid. This carbonic acid associates into bicarbonate and hydrogen. This is a slow process. Why? Because there is no catalyst available in the plasma. And now let's see what happens to the fate of these hydrogen ions. Whenever hydrogen ions are made, our body does not like to leave them like that because they are acidic and they can cause trouble. So these hydrogen ions, they are combined to the plasma proteins, and so they are like buffered. There we know there are proteins in the plasma. These hydrogen ions, they bind to those plasma proteins. And now let's come to this bigger arrow. We are still on the carbon dioxide. So the third way in which carbon dioxide it travels, it is attached to the hemoglobin, which will be this. So let's first discuss about this. This is also the dissolved, which is traveling like bicarbonate. So some of it got turned into bicarbonate in the plasma. The rest of it will be turned into bicarbonate inside the RBC. This is not traveling inside the RBC, but bicarbonate will be made inside the RBC. So this carbon dioxide, it goes into the RBC. In the RBC it combines with water, and now we have an enzyme known as carbonic anhydrase. This carbonic anhydrase makes carbonic acid, which associates into bicarbonate and hydrogen ion. And this is fast reaction because of this catalyst. And now let's see what happens to the bicarbonate ion. This is beautiful. This bicarbonate ion, it moves out of the cell. It wants to travel in the plasma. It does not want to travel in the RBC because this is the form of carbon dioxide, 70% of the carbon dioxide, which travels dissolved in the form of the bicarbonate ion. So it wants to go out in the plasma. So it moves into the plasma. And now what happens, that one anion has left the red blood cell. So as a result, the chloride, it moves into the red blood cell, and this is known as chloride shift. You have to know what is chloride shift. Chloride shift is that when bicarbonate ion, it moves from the RBC into the plasma, the chloride, it moves in the RBC because chloride is also an ion to maintain the electrolyte balance inside the RBC. This is known as chloride shift. Now, let's see what happens to the hydrogen ion inside the RBC. Like I said before, our body does not want to leave the hydrogen ion unattended. Hydrogen ion unattended can be dangerous. So this hydrogen ion, it combines to hemoglobin. Hemoglobin is also a buffer. It attaches to the hydrogen ion, and now this hemoglobin is said to be reduced hemoglobin, and it is written like HHB. I already in the previous slide you saw it written like this. So HHB is the reduced hemoglobin. So just remember that whenever the term reduction comes, it means either the addition of a hydrogen ion or removal of electrolytes or removal of oxygen. So this is reduced hemoglobin. And then now let's move to the other form of the carbon dioxide. We know that some of the carbon dioxide, it also attaches to the hemoglobin. So this carbon dioxide, when it comes into the red blood cell, it attaches to the hemoglobin. And where does it attach to the hemoglobin? It has its own designated spots. both carbon dioxide and oxygen, they have their own offices inside the RBC. They do not sit in each other's spot. Carbon dioxide, it attaches to amino acid in the globin chain of the hemoglobin, and now the hemoglobin is known as carbaminohemoglobin. This is how it is written when you see HbCO2. It means carbon dioxide is attached to the hemoglobin, but it is attached to the globin chain, to the amino acid in the globin chain, whereas oxygen, it is attached to the heme group, iron in the heme group. Okay, so do not confuse these two. All right. So now this is important thing is this, that when there is carbon dioxide inside the RBC, that carbon dioxide, it causes displacement or the unloading of oxygen from the hemoglobin. This is known as Bohr effect, right? This is known as Bohr effect. What is Bohr effect? When there is increased partial pressure of carbon dioxide, it causes unloading of oxygen from the hemoglobin, and this is known as Bohr effect. And this hemoglobin, it attaches uh, to the H, this hydrogen ion, and it is reduced, and this oxygen, it is released, it is unloaded from the hemoglobin, and it goes into the this interstitial spaces, and ultimately into the tissue. So this is how hemoglobin is unloaded at the level of the tissues. So if you want, you can revise all the factors which cause unloading of the hemoglobin and unloading of the oxygen from the hemoglobin in this slide, but I will not go over them again. Okay, let's quickly go over. So what happens in the pulmonary capillaries, right? Right now, we just saw a slide which was at the tissue level, at the systemic level, in the systemic capillaries. Now, what is happening in the pulmonary capillaries? Pulmonary capillaries, the opposite is happening. The bicarbonate, it moves from the RBC, while chloride moves out of the RBC back into the plasma, and then bicarbonate, it binds with the hydrogen ion to form H2CO3. Then H2CO3 moves in the other direction, and carbon dioxide is made, and that carbon dioxide diffuses into the alveoli. Let's see it in in the diagram so it can be more clear. Now let's see what is happening at the level of the lungs. Let's orient ourselves. This is alveolus, and this is pulmonary capillary. Why am I calling it pulmonary capillary? Because it is present at the level of the lung, with the alveolus. So how is it coming here? Pulmonary artery is coming from the heart, from the right side of the heart. That pulmonary artery has deoxygenated blood from the tissues. That pulmonary artery, it divides into smaller branches. Still at the level of the alveolus, it changes into pulmonary capillary. This pulmonary capillary is in direct contact with the alveolus. Now this thing is known as respiratory membrane. What is respiratory membrane made up of? So I'm starting from the pulmonary capillary side. So these are thin endothelial cells, which are squamous cells, and then there is a fused basement membrane, the basement membrane of the alveolus, and the capillary is fused. Then we have thin squamous cells which are lining the alveoli. So this is respiratory membrane, and it is very thin in normal healthy lung. In diseases, when it becomes thick, which can happen due to fluid in the lung. So if there is fluid on this side, it will thicken it, or if there is scarring of this membrane, it also becomes thick. So for any reason, if it becomes thick, this exchange of gases, it cannot happen to the amount that we want them to happen. Alright, so this was just a recap. This is respiratory membrane. Now let's come to our current topic. So we already said that carbon dioxide, it is transported in the blood in three forms. Number one form, it is transported in the blood in dissolved in plasma. So this is the carbon dioxide which was dissolved in plasma. Now it has reached the lungs from the systemic circulation, from the systemic capillaries. It moved into the right side of the heart, and then from the pulmonary artery it came into the pulmonary capillary, and now it has reached the lung. Now this carbon dioxide, it diffuses to the alveolus, and from here it is expired. And then the second portion is the one which travels as bicarbonate. So both this thin arrow and this thick arrow, This is the carbon dioxide which is traveling as bicarbonate. So this we know that a small portion it changes to bicarbonate in the plasma because there is no catalyst enzyme present in the plasma. So in the plasma the reaction is very slow, but some amount of carbon dioxide it does change into bicarbonate in the plasma. And like I said before, this reaction goes both ways. So when it reaches the lung, so this hydrogen ion and bicarbonate they combine and they make carbonic acid. Carbonic acid is then dissociated into carbon dioxide and water, and this carbon dioxide it diffuses across the thin respiratory membrane into the alveolus to be expired. All right, and now the second portion of this bicarbonate, remember these thin arrow and thick arrow, these are both the second point, the carbon dioxide which is traveling as bicarbonate ion. So what happens inside the RBC? So when they reach the lung, this bicarbonate ion it moves back into the RBC because it knows that the RBC has carbonic anhydrase and I can quickly be turned into carbon dioxide. And when this bicarbonate ion comes inside the RBC, it wants to maintain its electrolyte balance. So the chloride ion it moves out. Chloride shift is out, and this happens via a transport protein is present for this chloride. So this chloride comes out and this bicarbonate it comes in. When it comes in, it attaches to this hydrogen ion and it makes bicarbonate ion. Now this bicarbonate ion is catalyzed by the carbonic anhydrase and it changes into carbon dioxide and water. Notice that carbonic anhydrase is the only enzyme which is doing this, and it is working in both ways depending upon the on the location and the situation. So this carbonic anhydrase it changes this by weak H2CO3 into CO2 and H2O. This CO2 dissolves in the alveolus to be expired. So this is the second mode of transport of carbon dioxide. Now we said the third way in which the carbon dioxide travels in the blood, it is attached to the hemoglobin, and there is a separate seat for carbon dioxide and there is a separate seat of oxygen. This carbon dioxide it attaches to the globin chain, to amino acid of the globin chain, and it is known as carbamino hemoglobin, and this is how it is written. So we are done with the transport of carbon dioxide. Now let's see what is happening to the oxygen at the level of the lungs. At the level of the lungs, the oxygen needs to be moved into the blood. So the deoxygenated blood can be oxygenated and taken to the pulmonary through the pulmonary veins into the left side of the heart to be distributed in the body. So this oxygen comes in when the partial pressure of the oxygen it increases. Over here the partial pressure of the oxygen is now increasing in the lungs. The affinity of the hemoglobin for the oxygen increases. The hemoglobin it gets rid of the hydrogen ion and it attaches to the oxygen, right? And this is known as Bohr effect. What is Bohr effect? When the partial pressure of the oxygen increases, the affinity of the hemoglobin for the oxygen it increases, and hemoglobin it attaches to the oxygen. And now this hydrogen ion it is removed, and this hydrogen ion it will be used over here to make H2CO3. So this is how our blood it gets oxygenated in the pulmonary capillary and how it gets rid of the carbon dioxide. I went over Bohr effect. What is Bohr effect? Bohr effect is that when there is increased partial pressure of carbon dioxide, more carbon dioxide enters the RBC, and that carbon dioxide it results in displacement of oxygen from the hemoglobin. So increased partial pressure of the carbon dioxide it displaces oxygen from the hemoglobin. This is known as Bohr effect. There is another term which is known as Haldane effect. What is Haldane effect? Haldane effect is that the total amount of carbon dioxide transported in the blood is affected by the degree to which the blood is oxygenated. So in another word, the lower partial pressure of the oxygen it leads to lower hemoglobin saturation, and this lower hemoglobin saturation it means that the hemoglobin is reduced. When the hemoglobin loses oxygen, we say the hemoglobin is reduced, and now the blood can carry more carbon dioxide. So in simple words, the lower partial pressure of oxygen, blood can carry more carbon dioxide. This is known as Haldane effect. Now let's go over this diagram. In this diagram, so when the carbon dioxide, it affects the Bohr shift, and also it is going to affect explain the Haldane effect, because these things are kind of interrelated. So when carbon dioxide it enters the red blood cell, it makes H2CO3. That H2CO3, it is dissociated into H and HCO3. This H, the hydrogen ion, it combines with the hemoglobin. Now this is the reduced hemoglobin. This is the reduced hemoglobin. This reduced hemoglobin, it has more affinity for the carbon dioxide. So reduced hemoglobin has more affinity for the carbon dioxide. Now the blood will carry more carbon dioxide when there is reduced hemoglobin present. So the dissociation of oxygen allows more uh, carbon dioxide to combine with the hemoglobin. So this I will change correct this. This 2 is before, should come after the carbon dioxide. So this is our Haldane effect. Then that this reduced hemoglobin, it has more affinity for the carbon dioxide, and the oxygen, it is unloaded from the hemoglobin. So this oxygen is unloaded and this hemoglobin attaches to the H. This is known as Haldane effect. And then this is our Bohr effect. And this, just as a reminder over here, this is the chloride shift. So in this slide, it says just what I went over. So I will not go over this again. This carbonic acid bicarbonate system, it is a buffer system. What are buffers? Buffers are the substances which maintain the pH of the blood, right? And this bicarbonate, it is considered the alkaline reserve of the carbonic acid bicarbonate buffer system. So what happens? How does this system it help in resisting the change in pH? I'm going over this because in the next week we are going to talk in more detail about the acid base balance. So then we will come to know, we will talk more about these buffer systems. So what happens if the H plus ion concentration it rises in the blood? It means that the blood pH is acidotic, because when H plus the hydrogen ions they increase, the pH it decreases, and we say that this is there is acidosis. So when H plus concentration it rises in the blood, excess of the hydrogen ion it is removed by combining it with bicarbonate to make H2CO3, the carbonic acid, as we already saw this reaction. And what happens that this H2CO3 dissociates into carbon dioxide and water. And similarly, if I will talk about in next slide, then this carbon dioxide is removed, because when carbon dioxide is being removed, actually we are reducing H2CO3. And what happens if the H concentration it begins to drop? It means that the pH is going towards the towards alkalosis. So what happens that this H2CO3 it is dissociated and it releases H plus ions, and this H plus ion it brings the pH back to the normal. H2CO3 is considered the alkaline reserve or carbonic acid bicarbonate buffer system. You have to know this. Since we are doing the lungs, and lungs they play a very important role in maintaining the blood pH. Let's see how they do it. So changes in the respiratory rate and the depth they affect the blood pH. How do they do it? That slow shallow breathing it causes increase in carbon dioxide in the blood. If the respiratory rate is slow and the breaths are shallow, it means that too much carbon dioxide is not being removed from the body. As a result, there is increase in carbon dioxide in the blood, and this increase in carbon dioxide in the blood it results in low pH. Why? Because carbon dioxide, just consider it another form of H2CO3 bicarbonic acid, and that bicarbonic acid it brings the pH lower. There is acidosis, and what happens in rapid deep breathing, it causes decrease of carbon dioxide in the blood, because now the lung is removing the carbon dioxide, and that results in a rise in pH. And I want to state a small point here, that this rise in pH, maybe it is not because of carbon dioxide, maybe it is because of some other acid, like the lactic acid or the ketone bodies. But how lung is going to happen, going to help, because the basic end result the body is looking for, it wants to balance the pH in the blood. So what happens when the pH maybe the pH is high because of the metabolic reasons, but the lung, it is going to get rid of the carbon dioxide, and in this way it will decrease H2CO3, and the pH will be balanced. It will try to balance the pH. All right. And then, so this is the point. And the next point, changes in ventilation can help adjust pH when disturbances are caused by the metabolic factors, right? So in this way, the breathing and the respiratory system, it plays a very important part in maintaining the acid-base balance of the body. Now let's talk a little about hypoxia. We also discussed this topic of hypoxia when we were doing cardiovascular system. So hypoxia means that there is inadequate oxygen delivery to the tissues. It does not necessarily means that there is inadequate oxygen in the blood. It means that the oxygen which is reaching the tissues, that is inadequate, and it can be due to many causes. There can be anemic hypoxia. Where anemic hypoxia, the name is telling us that there are too little RBCs, or RBCs are present, but there is abnormal or very little hemoglobin. We went over these points when we did cardiovascular system. When we did our blood, sorry, not the cardiovascular system, and we were doing blood. And then there is ischemic hypoxia. What is ischemic hypoxia? For example, just consider that there is this vessel, right? This vessel, it is taking blood to the tissues. Now because of some reason, this vessel becomes clogged or blocked or narrowed. Right now, the same amount of blood, it cannot go to the tissue, it cannot pass through this vessel. So this is known as ischemic hypoxia, when there is blockage to the blood circulation. Then there is histotoxic hypoxia. What is histotoxic hypoxia? There are certain metabolic poisons. For example, cyanide. Cyanide, what does it do? It actually acts at the level of the respiratory chain. It at the mitochondria. It can prevent the utilization, the final, the final utilization of oxygen inside the cell. So it is a metabolic poison. The cells, they're unable to use the oxygen because of cyanide poisoning. Then there is hypoxemic hypoxemia. Hypoxemic, emic should tell you that this is the decreased amount of oxygen in the blood. Okay, so abnormal ventilation, pulmonary disease, low levels of oxygen in the air, they can all cause hypoxemia, hypoxemic hypoxia. And then because when there is abnormal ventilation, there is pulmonary disease, there is low level of oxygen in the air, all of these will result in decreased oxygen in the blood. And then there is carbon monoxide poisoning. And carbon monoxide poisoning, it can happen for many reasons, and hemoglobin has 200 times more affinity for carbon monoxide than oxygen. And these people, they have like very cherry red blood. And then remember, we talked about hyperbaric chambers, which applied the Henry's law. So these patients, they are put in the hyperbaric chamber to remove the carbon monoxide. So the person can again attach to the oxygen and get rid of the carbon monoxide. Victims have headaches and they become very flushed. It is kind of cherry red color. The carbon monoxide gives cherry red color to the blood. So this is all about hypoxia. And this is the end of our respiratory gas diffusion transport.